The RV254 Search-10 study is a successful collaboration between the U.S. Military HIV Research Program and the Thai Red Cross AIDS Research Center. We worked together from the inception of the study to the conduct of the study and now the analysis and reporting of the data. First of all, this is a study that we screen people in real time for acute HIV infection. We enroll people very fast from the time the client walks into our clinic to confirmation to enrollment is only 3 days. We then consent them to the main protocol, and also we have optional procedures that have been very well received by our volunteers. We also offer treatment as part of another protocol that we randomize people to 3 drugs versus 5 drugs, and the time from when we enroll people to performing the procedures to starting treatment is only 2 days.
This study, Search-010, is particularly important because what it allows us to do is to look at events that are happening at the very beginning of infection, earlier than any other previous studies. And so what that allows us to do is to say, what's happening at the various different places in the body? What is the virus doing? What are the cells and the immune system doing? So that we can hopefully identify ways that we can intervene. Number one, we can possibly identify ways that we can exploit the knowledge that we gain from this study to make better vaccines, because ultimately that's going to be the best hope for prevention. The second thing that we can do is we can understand what are those key steps in the series of events that happens from infection until, unfortunately, ultimately death in the untreated HIV-infected patient. What are those steps and how can we intervene so that the disease is less severe, or possibly we can even effect a functional cure?
For getting the acute HIV infection, first of all, the specimen, plasma specimen will be tested at anonymous clinic at Thai Red Cross using the high sensitive ELISA. For a single of specimen over there will send to us every day, then to do the 2 methods. For the first method, we do the low sensitive ELISA again, and if the results show non-reactive, we confirm the acute infection with the NAT again. If the NAT is positive, we can call that patient got acute HIV infection. And for the second method, we use the strategy named pool NAT. For this method, we will screen all the negative results from the high sensitive ELISA by NAT. We pool the specimen together and do the NAT. If the NAT show reactive, we break the pool to figure out which client is got HIV infection.
The majority of our patients with acute infection in Thailand and Bangkok are young men who have sex with men. They've been infected for about 2 to 3 weeks. They are mainly highly educated and they have about 3 sexual partners in the last month. The majority also had symptoms of acute retroviral syndrome. They have fever, they have aches and pains and fatigue, oral ulcer, skin rash. This is an opportunity for us to also diagnose acute infection in people who have these symptoms.
From April 2009 to October 2013, we screened over 87,000 people from anonymous clinic with 2 methods. We can get 158 clients who got acute HIV infection and 130 people enrolled in this study.
I think the unique thing about RV254 is based on the commitment of our clinical people and also on the commitment of our participants, that we're really catching these people early. And I think, as far as I know, this is unique in the world, that you really aim to catch people that early. Because normally if the scientific community is talking about acute HIV infection, it can be up to 4 months out of infection. But RV254 is really aiming to get people within days of infection.
We wanted to see what happened after early treatment in terms of HIV reservoir size. And here we look at the total HIV DNA in 1 million cells in the peripheral blood. And we compared our patients, the SURG10 RV254 volunteers, to two groups of people. One are the elite controllers. These are people who naturally can control virus without ever being on ART. And another group are people who have been on treatment for 5 years but started treatment late during chronic infection. And you can see here that after 1 year of treatment, our patients have similar reservoir size as elite controllers. We also were interested in looking at infection of the central memory CD4 T cells, and these are cells that are major barriers to HIV cure. They're long-lived, latently infected cells, and here we look at the amount of integrated HIV DNA in 1 million cells, and we compared 3 groups of people. The group in green that started treatment within the first 15 days, the group in blue that started treatment between 15 to 25 days, compared to in purple, people who started treatment late. And if you look at the slides here, you can see that in people who start treatment early, they had very low infection in the central memory CD4 T cells even at the beginning, and this remains undetectable after treatment. In people who start treatment during acute infection, but about 15 to 25 days after infection, they had detectable infection in the central memory CD4 T cells, but again, after 24 months, the level of infection is extremely low. In contrast, people who start treatment late, even after 2 years of treatment, they remain with high infection in these cells, and this will remain for many, many years, and it's a major barrier to cure.
In the context of the SURG10 or RV254 study, we are particularly interested in the immune response and the development of the cell populations within the mucosa of the RV254 participants. We are thinking that this is very interesting because the first entry portal of HIV when a human is getting infected is the mucosa. So we try to understand what is happening in acute infection in the mucosa to get a better idea how we can, prevent infection in terms of a vaccine, or even to kind of, make antiretroviral treatments better.
Why is AFRIM's Department of Retrovirology interested in an acute infection study? And the reasons really are that we are a vaccine-focused organization. So we're an Army organization, and the Army has a program that has said to us, we need you to develop a globally effective HIV vaccine to protect the warfighters, our troops, and to protect other people in the world. So with that as our mission, we are looking for ways to not only develop products way at the far end of the development spectrum, we're also looking for ways to understand the science behind HIV infection so that we can identify new opportunities to stop those infections.
We've been fortunate to work with experts at the MHRP and also many collaborators around the world, and we've successfully published papers on the effect of early treatment, on the reservoir size and immune preservation, as well as to document what happens in acute infection in the central nervous system, as well as propose a new staging for acute HIV infection. And we have many more publications coming in the next few years. Our work was selected as an oral presentation at the Conference on Retroviruses and Opportunistic Infection in March this year in Atlanta, and also for the press conference. And since that conference, we've had quite good news coverage of our work. Our work was featured in the Journal of Science in the news section. Our work was covered in AIDS Map, and the Thai press has also picked this study up, and we were invited to appear on television channel, and also our work was covered in the Thai newspaper. This work has really generated a lot of interest in the country and internationally on trying to get people in to early diagnosis of HIV and early treatment, and there's hope that this will eventually lead people to be able to achieve HIV cure?
There are a couple of things about being in Thailand that make this partnership between AFRIMS and Search uniquely fruitful. And the first is there is a favorable disposition on the part of the population here in Thailand who become HIV infected. They want to help. They're very motivated to be participants in research to try and help other people who are suffering from that same disease that they suffer from to benefit. And so that's one thing, is the favorable disposition towards participation in research. The other thing that is very notable and is much different than some other places I've worked is the staff, both at AFRIMS and at Search, are extremely dedicated and extremely loyal. They tend to stay in place for a very long time and develop years of a depth of expertise in their relative fields. So I think those two things underlie the unique fruitfulness of this partnership between AFRIMS and Search.
Participants are really involved in these studies, so they really want to make a difference. So they're not in it because they're getting a little bit of money, which is part of every trial. They really want to help the scientific community to find a cure against HIV. And I think that's very unique about Thailand.
So with regard to optimism regarding an HIV vaccine, there's a great battle, really, between the virus and our efforts to prevent it. The virus is extremely good at evading the human immune system. It's adapted to do exactly that, to wipe out the very cells that are best at fighting it. So they take out the very best fighters at first and then infect all the rest of them. That's a great challenge. And because of that challenge, every vaccine that we've ever tested to see if it prevents HIV infection has failed. On the other hand, there is a vaccine that was tested here in Thailand with a consortium including a vast array of collaborators in a study called RV144 that demonstrated that 31.2% of people who received the vaccine were protected. So there was a vaccine efficacy, finally, of 31.2% at 3.5 years following vaccine administration. So that gives us hope that it is possible. It is possible to have an HIV vaccine, but this is only the start, because 31% is not enough to probably have the impact that we want to stop the pandemic around the world. This also is a regional vaccine, so it's only designed to work in Southeast Asia, and it may or may not work in other places. The message that I would leave, though, with the viewers is that there is cause for optimism, because there is a vaccine that worked partially. We have clues as to how it worked, and we have strategies to try and capitalize on those clues moving forward in product development.
So if we look at the steps to HIV cure, we can say that the first important step is to diagnose people early. The second step is to institute immediate treatment to limit infection of the latently infected cells. And then we could give additional interventions such as therapeutic HIV vaccine or neutralizing antibody. And then we hope that our patients will achieve functional cure, which is being able to control virus without lifelong antiretroviral treatment.
แน่นอนครับถ้าเกิดว่ามีการวิจัยอย่างอื่นในอนาคตนี้ผมคิดว่าผมจะเข้าร่วมแน่นอนครับสิ่งหนึ่งที่ทำให้เรารู้สึกว่าตัวเองยังมีค่าอยู่ก็คือการได้มีประโยชน์ต่อผู้ติดเชื้อคนอื่นด้วย